So Bruce, I I have a project. I've got B cells. I got to analyze them. I got to start from scratch. But I got to tell you, man, it can't take as long to build this model as it has in the past. So what do you got for me?
Well, we've been really working hard on that. We're trying to make it so that it's easy to use. And so why don't I take you through it
That'd be
and
good because I got work to do, man.
okay, fair enough. All right. So I'm going to launch our new GemStone. This is going to be the beginning of GemStone 2. It's a new version. And the first thing that we do is we'll load in a B cell file. It's part of a project that we're doing. And we'll load in three important measurements for selection of B cells.
Cool.
So it'll be CD19, side scatter, and CD22.
All right.
Now, one of the things that we've learned in doing these kinds of models is that when you have a case where the expression that you're interested in ends up being really complicated, which is the case for CD19 and CD22, that the best strategy is to first go after the negatives and then create a kind of inverse type of model that will get the CD19 positive and CD22 positives. This is something that we've done on a number of models, and so I'll show you this strategy right now.
Excellent.
Okay, so we'll expand CD19, and in this case we're going to be looking for the CD19 negatives. So we choose a constant profile for dimmest peak. And then here's something new with the new system is that we'll choose what we call a low pass type of an expression profile. And we call it a low pass because it acts kind of like a filter
Mm-hmm.
where it's only going to be accepting events that are low, no matter how complicated they may be. And you'll see that when we go after the CD19 positives, we're going to take the inverse of the low pass so we can get CD19 positives without actually finding a peak and so on for CD19.
Okay.
Okay.
So it's easier in this case to figure out what the B cells are not to get started.
Exactly.
Okay.
So that takes care of CD19. And now we go down to light scatter. Now, light scatter is interesting because there's usually a lot of overlap with light scatter. So we usually have to tell our peak finding system to look for peaks using the steepest side of the peaks. This comes all the way from DNA analysis, where if you want to find the G0, G1 standard deviation, you look at the steepest side of the peak. So that's what we're going to do here with side scatter. And we just come over to the peak, icon changes, and we'll choose steeper side. And once we've set that up, we can then go ahead and choose the dimmest peak, and we're done with side scatter.
Okay, that was pretty easy.
So we have CD19 negatives that are lymphocytes at this point. And then we come down to CD22, and it's the same strategy. We choose the lowest expression profile here, and we also are going to change this one, so it's going to be low pass. And at that point, we're pretty much done with the CD19 negative part of the problem. We just need to change the name from cell type to something that's meaningful. So we'll go down here to edit common parameters, and we'll choose, we'll type in CD19 negative here. And as soon as we do that, you'll see the tab changes to CD19 negative. And then we do duplicate cell type. And when we get this question here, what it's really asking you is, okay, I'm going to go ahead and duplicate this cell type for you, but do you want me also to bring all the expression profiles over to the duplicate cell type? And do you want them all to be linked together so that if I change one location, it's going to change the location in the other cell type?
Okay, and that seems like something we would want to do in this case.
Exactly. So we go ahead and we say yes, and then this is a little ease of use feature that we've added. We can now just change the name right now of the duplicated cell type to CD19 positives. And now you can see that right now it's duplicated, so
Yep.
this is actually not what we want. We want the inverse of this. So we go to our little pencil icon here. Instead of low pass, we choose inverse low pass. And we go down to our CD22 and do the same thing.
Okay, and I can see it's selecting things that are outside of that lower band now. Okay.
Right, and if we want to just take a look at what's really happening here, we can create a 1P distribution, and I'm going to go in and edit some properties here. So I double click, choose unclassified. I don't want to see the unclassifieds, but I want to see the CD19 negatives and positives, so I turn on this option.
So the gray portion are the negatives and the blue positives. Okay.
Yeah, and the key strategy here is even though it's finding a peak, you can see it with this little black triangle, it doesn't actually have to find a peak with this strategy since it can lock in on the CD19 negatives.
Okay.
And we have found that for the B cells, this ends up being a very robust type of modeling system that just allows us to automate things that we weren't able to do before doing this.
Sure.
So we're actually done with this part of the tutorial. The next step is we want to save the model, and then we want to try it on some other samples that are actually from different institutions using different panels for B cells.
Well, that would be great, because that's what I've got coming up to me.
Okay, so we'll go File, and we click on Save Template As. We'll choose B cell 1. We'll replace this. And then the next part is that we want everything to be automated. So we're going to tell it, we're going to do a little programming here. We're going to change these files, which are B cell files. We'll multiple select these and put them on a different level.
Okay. This is an outlining type level you've got here.
Right. And the neat thing is that each level can do different things to the files. So in this case, what we want, we'll just scroll down here, we want the system to load in our template file that we just created. So we just go ahead and choose that. And we say OK. And then we wanted to do an automated analysis on it, which is checked, so that's what it's going to do.
Okay.
So at this point, the system's ready for us to automatically analyze for B cells in bone marrow. To do that, we just double click on the file, and we'll go through the same strategy that we just did, and you can see it has captured the CD19 positives there. But now we'll go down to another file, which is completely different, comes from a different lab, and you can see very different location for CD19. The peak's not, it's still catching the peak, but you can see that it's quite possible that it might not actually see a positive peak, but it's still going to work. And then we'll do a third file here. And
Okay.
that's all there is to it.
Well, that's going to help me a lot.
All right.
Thank
Well,
you, man.
okay.